In this activity, you will learn about the life cycle of a jellyfish and how it relates to the recent explosions of jellyfish populations, called jellyfish blooms. You will design experiments testing a jellyfish's ability to transition from the polyp to ephyra stage of development. You're going to read through the slides and/or watch this video with the background info about the life cycle of a jellyfish. Then you're going to pick one research question to study and design an experiment on the jellyfish. You are also more than welcome to coordinate with a classmate to work on this assignment together. If you choose to work with a partner, you both must submit the assignment into Google Classroom. However, it can be the same document that you both worked collaboratively on. So what causes jellyfish blooms? A jellyfish bloom is defined as a substantial increase in jellyfish populations within a short period of time, the result of a higher reproduction rate. So this is a picture that I took of two lion's mane jellyfish. Um, Actually, just this past Labor Day in Connecticut, in Niantic Bay. So they were currently undergoing a big jellyfish bloom over there. There were jellyfish everywhere, all in the water while we were out in the boat. So we could see these bright pink jellyfish just popping up all over the place. So, recent studies have been showing that jellyfish population blooms are more frequent and are continuously increasing in size. And jellyfish blooms are a problem. Um, Not only are they a threat to humans because of stings and they get caught in fishermen's nets and they make it very difficult for sometimes boats even to travel through the water. They are a threat to the natural balance of marine ecosystems. So this is the jellyfish life cycle. We start with some mature Medusa jellyfish. We have a male releasing sperm and a female releasing eggs into the water. The sperm will fertilize the eggs and it will develop into a jellyfish larva. That jellyfish larva will float through the water and it will eventually settle on a hard surface called a substrate. Once it lands on the substrate, that larva will develop into a polyp, so that vase-shaped cnidarian. And then eventually that polyp is going to grow and it will elongate and bud off young immature jellyfish called ephyrae. This is a little baby jellyfish here. The research focus to date has been on the Medusa phase of the life cycle, but the key to our understanding of jellyfish population lies in the polyp phase of the life cycle. The primary purpose of the polyp is to undergo asexual reproduction, or cloning, and to produce several microscopic jellyfish called ephyrae, which when released grow rapidly into adult medusa. A single jellyfish polyp can make many ephyrae. And so understanding how the polyps um, behave and what factors in the environment influence their development into ephyrae can cause the population of adult medusa jellyfish to grow. So the more healthy polyps you have, more ephyrae you have. Or if you have just a lot of happy polyps that are making a whole bunch of ephyrae, you're going to get a big jellyfish population. I strongly suggest you watch this video. I'm not going to play it here because you're not going to be able to hear the sound, but this video shows the polyp turning into the ephyrae and releasing all the little baby jellyfish. Your task is to design an experiment on a jellyfish polyp's ability to produce ephyrae. You're going to choose one of the following research questions. Then determine the independent and dependent variables. You're going to create an experiment and describe the steps you would take to carry it out. Consider your control and experimental groups. Think about what are some of the variables you need to control for, or the variables that you need to keep constant. You need to think about how you will collect the data. And then you're going to draw a picture of your experimental setup and upload a photo of your drawing or make some kind of digital drawing. For example, I might want to research a question like, do jellyfish polyps make the most fiery in acidic environments? So my independent variable, the variable that I'm going to change will be the pH of the water. The dependent variable I'm going to measure is the average number of ephyrae per polyp. So for my experimental design, I would take some adult male and female jellyfish and allow them to spawn and produce jellyfish larvae. I would distribute the larvae in 6 different saltwater tanks. Once the polyps formed, I would add acids to each tank to decrease the pH and make it more acidic. Then I would allow polyps to develop into ephyrae. I would count the number of ephyrae produced and divide by the number of surviving polyps to get an average ephyrae per polyp measurement. So some of the things that I would need to control for in my experiment, I would need to consider the amount and type of food I was giving to the jellyfish polyps. I need to maintain a constant temperature of the water and salinity of the water. I'd have to use the same size tank. I would have to make sure that the oxygen levels in the water was the same. I'd have to expose each tank to the same amount of light or darkness, and I'd have to make sure that the same substrate material was available for the polyps to attach and grow. And so this is my digital drawing. So I have 6 tanks. I have my control group, which is set to a pH of 8.1, which is normal seawater. And then I would have 6 tanks decreasing the pH by a little bit. And I would count how many ephyrae are produced in each jellyfish tank at each pH, and then I would have to divide by the number of polyps that I had in each tank in order to figure out how many ephyrae per polyp was produced. So these are questions for you to choose from. Polyp growth in light versus the dark. Polyp growth under different salinities, so different amounts of salt. Do artificial substrates provide increased habitat for polyps? How does temperature influence the asexual growth of polyps? How much cold and warm water does a jellyfish polyp need to produce ephyrae? Or how does the type or amount of food influence the number of ephyrae? So you're going to pick one of these 5 questions and design an experiment. So include independent and dependent variables, include a control and experimental group, think about some of your constants, and consider how are you going to collect measurements and collect data. And then some kind of image with your experimental setup. If you have any questions on this assignment, please send me an email.